My name is Ella Sherwatsky, and today I'm going to talk to you about how to taxonomically classify short reads. Now, I want to preface this by saying that there's very limited information, taxonomic information, that you can get from short reads. So take all of your results with a grain of salt, and don't expect to publish a Nature paper based on these kinds of tools, but it is useful as a starting point. We're going to learn how to use two tools that are designed to classify short reads. One of them is called Centrifuge, and that's based on the Burroughs-Wheeler alignment, just like bow tie. And it takes the nucleotide sequences. The other one is called Kaiju, and that one is based on protein sequences, so it will translate your short reads into amino acids and compare them to protein databases. Now, as you can imagine, usually reads are pretty short. They're 100, 200 base pairs long. Your average coding gene is about 1,000 base pairs long. So you're looking at 10 to 20% of that gene and trying to assign taxonomy by a region that may or may not even be taxonomically informative. So this is why these analyses are kind of, I would say, a little bit iffy. But again, it's a good place to start. So let's look at how you do this, and we're going to start with Kaiju because it has a convenient web platform that you can use to classify your reads without your own resources. So let me share my screen. Here we are. All right, so let's start with Kaiju. All you have to do is get on the Kaiju website, into the web server. And all you need to do is you pick a name for your job. For example, when I ran this before, I used this name to signal that this is for a BVCN tutorial using Kaiju. These are reads that I used in the Trimomatic tutorial, and they were from a site called Cat, which is unfortunately not a real cat. And then in this case, I'm only using the forward reads, so I called it R1. And then you can select the file that you want to upload, and it can be gzip. You don't have to unzip your files. Once you select your file, there will be a button here that will say Start Upload, and you need to click that to upload the file into the Kaiju server. If you're going to rely on short reads, and we'll see this in a minute, I do recommend using your paired reads, which means you have to do one of two things. A, you use the reads as you got them from the sequencing center without any quality control, or only by trimming by length to ensure that you have paired reads throughout. So you don't have any reads that are not paired. And I highly discourage you from doing that, because garbage in, garbage out, if you don't do quality control on your reads, then your taxonomic assignments will be equally bad. Your other option is to only use the paired reads, which is what I did here. So only reads where you have a forward and a reverse. So to upload the reverse, you would just click this checkbox, you would select a file and then hit Start Upload, and wait until it finishes uploading. Once you've uploaded your files, you pick a reference database. You have four options here. The first one is using RefSeq genomes. This is RefSeq from 2016, so it's not a very up-to-date version. I've tried to run it and the results were not good. I didn't get many taxonomic assignments, so I probably wouldn't do that. There's this database called ProGenomes, which I actually haven't heard of until recently, but it is pretty up to date, and it even has more newly identified phyla like the candidate phyloradiation in bacteria and Deep Anarchaea. And this is the database that I used in the results that I'm going to show you. I think it works really well. However, all of the first three databases will only give you bacteria, archaea, and viruses. If you have any eukaryotes in your samples and you want to get that taxonomic alignment, you're going to have to pick the last database. And we'll get to that in a minute. This third database is the NCBI non-redundant database. The difference between using NR and, let's say, RefSeq is, first of all, it's not curated, so the results are not as reliable. And the second thing is it has a lot of unclassified environmental samples. And so you're very likely to get environmental samples as one of your main results, which is not very useful. But it is a larger database than RefSeq, much larger. So, you know, it's really up to you on what kind of results you want to get. And then finally, if you have fungi or microbial eukaryotes in your samples, then you have to use this last option of the non-redundant database plus eukaryotes. Again, in this example, I've used the progenomes database. There are some run parameters here. I just use the defaults. It again depends on how accurate you want things to be. So filtering low complexity protein query sequences will increase your reliability. Using a greedy algorithm means that you allow mismatches so the reads can map to something that's not a perfect match. If you want exact matches, then a lot less of your reads are going to be classified. Just be ready for that. So it depends on, do you want reads that are not a perfect match because they're maybe from a different strain or a close relative, or do you not want to look at that at all? That depends on your analysis. And then finally, these parameters have to do with the scoring matrix and what's your threshold that you're willing to accept as a hit or a match between a read and a protein. So once you've picked all of these, you would hit submit, and I've already done that for you. So let's look at some results. First, let's look at taxonomic assignment of only the forward reads. So as you can see, it tells you which genome you used. This genome is from 2017. I'll get back to that in a minute. But a lot of things have changed since 2017, so I would, again, take things with a grain of salt. And then it tells you which run parameters you used. Next, it says, you know, almost all of the reads were classified. And then it gives you this nice bubble plot. The size of the bubbles indicates the abundance of the organism. And then the colors are for phylum. So we have a large group of proteobacteria. We have a lot of Verrucomicrobia, which is weird because they're not super abundant. This is a marine sample, so I would not expect this. Here we have some cyanobacteria that's expected. It says Nostocalis if you look at the pop-up text. That's unlikely. These are freshwater cyanobacteria, so I think this is the kind of classification that you can expect from short reads. Again, it's not going to be super accurate. Definitely do not trust it to the genus level. Like phylum, order, maybe family, definitely not below that. All right, so this is a nice chart, and you can download it as is if you want to use it in your publication. I find it not super informative, and I prefer this one, which is a corona chart or a sunburst chart. Let's see what that looks like. So this is a corona chart. It's basically an interactive pie chart. And it tells you have 8% unclassified sequences. Then the rest are cellular organisms. Almost all of them are bacteria. You do have some Euryarchaea here. If you want to look at the bacteria, you just double click here. And now we can look at the taxonomic composition of bacteria in this sample. So we see Here are the cyanobacteria. Here are some verruca microbia, a bunch of alpha protobacteria, which makes sense. So these are all in all pretty normal results for a marine sample. We have some flavobacteria here. They're very common, some gamma protobacteria. So it's not bad, but again, I wouldn't trust anything below family level classification here. You can download this, I think you can download the chronocharts now, actually. But if not, then you can just snapshot it right here. Okay. How does this compare to running the paired reads? So I've also done that for you. I accidentally named this R1, but these are paired reads, and you'll see in a minute. I use the same database, the same parameters. Before this, we had about 104,000 reads that were mapped. Now we have 110,000 of the read pairs that were classified. But the classification looks pretty different. So we have a lot of firmicutes here. Some proteobacteria, that's fine. Cyanobacteria, again, it says Nostocalis. So using the paired reads did not improve the taxonomic classification in this case. And once again, we can look at the chronochart. And it looks pretty similar. Let's click on bacteria. And it looks like we haven't gained a ton of knowledge here, except there are a lot more firmicutes than there were in the forward-only reads. And I would say less flavobacteria, probably. So, you know, these are nice results, definitely something that you can start with, and mainly very easy to run if you're willing to go sample by sample. If you have a lot of samples, Kaiju does have a command line interface that you can use. And then if you use that, you can also download newer databases that will work with it. So the online databases are all from 2017, but if you scroll all the way to the bottom, there are newer databases that are only for download for use with the command line version. And these are from 2019. And if you really want the latest, I think RefSeq just had a new update, March 6th of 2020. Okay, so that was Kaiju. It's really easy. The command line version is also super easy. If you're looking for a tutorial on how to use it, it is a part of the NVO pipeline. But it's also just really easy to use. So Kaiju was based on protein databases. Now let's move on to Centrifuge. Centrifuge, like I said, it works on nucleotide sequences. And the way it works, it's very similar actually to read aligners like Bowtie and BBMap. So it first builds an index of the database that you want to use, and then it aligns the reads to that database. It aligns nucleotides, not amino acids. So when you want to run Centrifuge, the instructions here are, I would say, medium friendly, but understandable. So there are a bunch of databases that you can download that are Centrifuge databases. I forgot to say, so Centrifuge, I'm going to show you the command line interface. I highly recommend installing it on a server, just because the databases are very big and they take a long time to download. So if you want to download it to your personal machine, make sure that you have a very stable internet connection and be ready for it to take a long, long time. So the way you download the databases, Centrifuge actually has functions for that called Centrifuge download. What this does is, first of all, it downloads the taxonomy based on NCBI taxonomy into a folder called taxonomy. And then it's going to download a database of, in this case, archaea, bacteria, and viral sequences, genome sequences from RefSeq. And it's going to create a mapping file called seq ID to tax ID. And what that does is it translates the NCBI taxonomic ID to something that's actually readable. All right. The way you would build an index, and then you download the FASTA or FASTA reference sequence, the actual genomes, and continue to build your database. What they're doing here is they're using 4 threads. So -p is the number of threads that you're using. You're giving it the conversion table from the seq ID to tax ID. You're giving it a taxonomic or phylogenetic tree for that database, names file, and the sequences, and then finally an alias for the database. In this case, ABV for archaea, bacteria, viruses. Once you've built your index, you can go ahead and align reads to it. What I'm going to show you on my server is I actually downloaded the non-redundant nucleotide database from NCBI, which is called NT. And I created a Centrifuge database from that. And there are also instructions on how to do this right here in the Centrifuge website. So before I go on to the command line interface, I'm just going to say one more thing, and that is that if you don't have a server, Cyverse also has Centrifuge installed. If you don't know Cyverse, it's actually a really useful platform for bioinformatics. I highly recommend it. It's app-based, so you don't have to be an expert in command line. You don't even have to know how to use command line to use it. So you would sign up for an account, and then it gives you a certain amount of memory. And you can run all sorts of bioinformatics. You can upload your data and run all sorts of bioinformatics analysis on it. The way you would do this, you double-click here on apps. And then you can search the apps here in the search field. So I'm going to start typing Centrifuge. It's going to search. And voila, we have various versions of Centrifuge here. 104 is the latest that they have. So you would click on this, double click on this. Hope that it works. It's supposed to work. My computer is working a little hard right now. There it is. Okay, so you would give your analysis a name. So for example, I like to leave the name of the app that I'm using, and then in this case, give it some sort of informational or informative tag, so BVCN. It asks you which folder you want to put it in. I just leave it as the default, which is going to be my username and then analysis. As an input, you can give it a file or a directory. So you would want your, if you want to give it paired reads, you want to give it a folder, and they have to be in the same folder. So you would browse and pick files. In this case, all of my stuff is in this folder called raw reads. And I'm going to pick this folder. And then you can choose parameters. So you can use your reference database. The default is bacteria, archaea, viruses, and human. But you can also use only bacteria and archaea, or the non-redundant nucleotide database from NCBI. And then you have to tell it what's the format of your file. In this case, I gave it FASTQ files. And I'm telling it that the reads are paired. This is a pretty cool parameter to use. You can exclude taxonomic IDs. So if, for example, I was using the NR database or NT, and I don't want to get results that just say uncultured bacterium or environmental samples, I can go onto the NCBI website, figure out what is the tax ID for uncultured bacterium and environmental sample. It's actually not that hard to do. And then I would just type these IDs here. And then I'm pretty sure they're just separated by a comma. And that's it. And then you would just hit launch analysis. And once you do that, it will tell you here, in this case, I already submitted it. As you can see here, I actually have tried running Centrifuge on Cyverse a couple of times and it failed. I don't know why. So now I'm waiting to see if the earlier version will work. But I'm pretty sure they'll fix it, because another thing about Cyverse is they're really open to suggestions and to problems. If you have any, they'll answer pretty quickly. So that's how you run Centrifuge on Cyverse. And now I'm gonna switch to my command line to show you how I ran Centrifuge in the command line. So here it is. Okay, so we looked at the Centrifuge website. It does tell you how to run the tool itself. All it takes is Centrifuge. You have this flag -x. -x is the database that you're going to use, or the index, right? In this case, I told it to use NT, which I already downloaded and built earlier. -U means I'm using single end reads. And that's because I'm only giving it the forward read in this case for the sake of speed. So I'm giving it -U and then the forward read. Again, it's zipped. You don't have to unzip your files. If I wanted to give it paired reads, instead of -U, I would write -1 and then the forward read, and then -2 and the reverse read. Then I'm giving it the name of the output file, and I am going to ask it to also provide me with a human readable report file named R1 and then Centrifuge report. I accidentally named it CSV. It actually comes out as tab delimited, but you can fix that later. And then the last thing I'm doing here is, once again, I'm using the flag -P to tell it how many threads I want to run. This is really important if you're using a shared machine, to limit the number of threads that you're using. So I'm using 10 threads here. And I already ran this, so I can show you. This is what the output file looks like. You have your read ID, then your accession number of the result of the hit. This is the tax ID of NCBI. So this is what you'd be looking for if you were looking for a tax ID of the environmental samples or uncultured bacteria and that kind of stuff. As you can see, it's just a number and it's really meaningless to read as is, which is why we gave it that mapping file that takes this ID and actually tells you what's the taxonomy. The next thing is that it tells you the score of the best hit, which is right here. Then the score of the second best hit. What is the length of the hit? So the overlap between your read and the genome that it matched. The query length, so that's the length of your read, okay? And then the number of matches. So how many genomes did this read hit? In this case, it hit 5 genomes. And as you can see, we have 5 results for it here. So this would be the read ID, right? So you get all 5 hits here. They actually all have the same score. So it's gonna be hard to pick how to classify it. And that's, again, the problem with classifying short reads. So this is really nice, but not super helpful. So let's look at the report that is generated. It's kind of hard to read here. So what I'm going to do is I actually downloaded this table and I'm going to show it to you in Excel. So let me open the table. And share my screen. Okay. So here's what it looks like. You have the name or the taxonomy. That's the same tax ID that we saw before. That is just some random number. The taxonomy rank. That means at what level is your read assigned a taxonomy. So it can be species, genus, class, order, phylum, etc. Here you have the size of the genome that the read was mapped to. And we'll see in a minute why that is. Here we have the number of reads that were mapped to that genome. And how many reads were mapped only to that genome. So this is the number of unique reads. And as you can see, it's much smaller than the number of reads that map to that genome. What that means is that most reads will map to multiple genomes. And that makes sense, again, because you can't get very accurate taxonomy with short reads. Now, why did we need the genome size? That is because if you have a bigger genome that you're mapping reads to, you can get a lot more reads mapping to it just because the genome is a lot bigger. So if you want to know what your coverage of that genome is, which is a measure of abundance, then you would actually divide your number of reads by the genome size, and that gives you the coverage of that genome in your sample. In this case, I actually sorted this table by the number of reads. So you have a lot of reads hitting uncultured bacterium. The genome size is ridiculously large and makes absolutely no sense because it's probably a collection of a lot of bacteria. And the coverage is really low. If I was sorting by abundance, if you ignore this weird first row, which I don't know what always shows up first, these would be your most highly covered genomes. And those make sense. You have some alpha proteobacteria, you have cyanobacteria, Synechococcus, a Micromonas, which is microbial eukaryote. All of these make sense in a marine sample. Taxonomy is not super informative again, but this is what you can get with this database. If we were using RefSeq, perhaps we'd get somewhat more informative results, but they would be more limited just because the database is a little older. So that's it. This is how we use Kaiju and Centrifuge to determine taxonomic assignment for short reads. My favorite use of these tools is actually not for short reads, but for short contigs. I would rather assemble my sample and let's say pick all the contigs that are bigger than 500 base pairs or 1,000 base pairs, and that will generate a more reliable and more informative taxonomic assignment just because the sequence is longer. And yes, some reads may be lost along the way, but if their taxonomic assignment was not super reliable to begin with, I'm not sure what we're actually losing by that. So it's up to you. Do what fits with your analysis. In any case, I hope this was helpful and I'll see you in the next tutorial. Bye.